Hello everyone, welcome to sqadia.com. I'm Hina Khan, and today the topic of our lecture is poisoning and the pathologist. Now, how do we define a poison? It can be any substance which is injurious, which is causing harm, or even it can cause death of the organism. The sources usually include the animal and the plant sources that we talk about. And then how is a pathologist going to deal with such cases? How is he going to basically diagnose the type of poisoning that has the foul play that has taken place in different types of cases and in different types of autopsies that he's going to encounter in his or her life? So the first section basically deals with the poison and the autopsy in general. So the terminologies which are used while we are dealing with the forensic toxicology, that is basically the branch of forensic medicine that deals with the poisons and their injurious or harmful effects on the human body, that are discussed over here in detail. Moving on, the sources and the incidence of poison. Again, the animal and the plant sources are the mainstay over here. And how are we going to incidence, that is, whether describe them as a suicidal, as accidental, or iatrogenic type of poisoning that are discussed in detail over here. Now, talking about the autopsy in suspected poisoning, it is again very important how are we going to conduct the autopsy, what is the whole procedure, and the sample collection is the mainstay whenever we are talking about the toxicological autopsy that is being carried out. In the second section is the time of sampling. It is very crucial to attain the time of sampling. What was the time since death and how much time has elapsed since the sampling is done. It is very important. The information supplied to the laboratories for after the sample collection is again very important. How do we label these samples in detail? It is very important. The containers and the preservatives used in different types of cases, they are discussed over here in this second section. The third section mainly deals with the sites and the techniques of the sampling. What are the conventional sites and the sampling techniques that are being done, as well as the unconventional methods. The bile, the urine, the blood, the CSF, that is the cerebrospinal fluid, is discussed in detail over here. The vitreous humor, stomach, and the intestinal contents are very crucial and they have their own significances. The vomitus and the other fluids are also sometimes sent to the chemical pathologist if available at the autopsy. Now, the fourth and the final section basically deals with the volatile samples and the body tissues. Volatile samples basically deals with the gases, the gaseous materials, for example, the carbon monoxide. So the blood tissue sampling in such cases, how is it done? And the injected substances, for example, the morphines, the narcotics, and the drugs of abuse, they are discussed in this fourth section. I hope you want to learn more about this poisoning and the pathologist. Do watch the other sections to this lecture as well.